Okay, moving on. Chemotherapy that has been better at working, maybe without the baldness. And this is really interesting. Chemotherapy works fine, but it would be better without the baldness, maybe even without the nausea. Researchers at Georgetown University say they're one step closer to figuring out how to reliably kill malignant cells without damaging any of the healthy cells. And one of the secrets, it's a new technique that allows doctors to grow cancer cells and healthy cells together in the lab and to keep them from dying. Next, test drugs on a patient's tissue in the lab to see which ones kill the bad cells, but then leave the good cells intact. Now, the interesting thing is that that's out at Georgetown. Here in Long Beach, oncologist Dr. Robert Nagourney has been personalizing chemotherapy for years, often challenging standardized chemo protocols. And so he is joining me now in studio to talk about the latest breakthrough in the whole idea of personalized chemotherapy. And when this came out, this latest journal and publication, did you feel a little bit vindicated because you have been taking this approach for so long?
Yes, I would say I did. I'm particularly impressed that these people recognize the importance of personalized care and the concept of individualized treatment, and also that they're looking at whole cell experiments, that they're looking at the cell as opposed to the gene or the protein extracted from it.
Well, it does make sense that your approach and the approach that they are talking about at Georgetown, being able to actually work on a cellular level and then somewhat personalize the treatment for that type of cell, makes sense. Why do so many other doctors and hospitals just follow a standard protocol?
We've never really been able to do this very well, not until the last few years. And there were a couple of fundamental changes, one of which is we realized that cancer cells often don't grow too much, they die too little. So you have to capture cells in a way that kills them and doesn't stop them growing.
Say that one more time. They don't grow too much.
Cancer cells don't grow too much. They die too little. It's a process of immortalization. And so learning how to study cells correctly has been the breakthrough that's enabled us to pick treatments for our patients.
Now, doctor, I would imagine that you have your detractors, because if your personalized chemo cocktail worked 100%, I would imagine every doctor in the world would be following what you do. How much better, if at all, is your success rate over standard protocols?
On average, we've approximately doubled the response rates. We've just submitted a paper in lung cancer. Really? We've just submitted a paper in lung cancer. We've also shown that in breast, ovary, and other diseases. Now, remember that most patients only respond 20 or 30% of the time. So a doubling might be 40, 50, 60, 70%. We're never perfect.
But that's huge.
We think so.
Huge.
We think it's huge for the patient.
Now, you take out the cancer tumor, and then you actually say it stays alive for 4 or 5 days, and that allows you to do experiments on those cells. Is that correct?
Yes. All cancer cells will stay alive for a few days in tissue culture. What's different about this particular paper is that they immortalize them and keep them alive forever, presumably. Okay. We don't necessarily feel that's an advantage, because we want to capture cells with a snapshot, a quick look at what they're going to respond like, and that's the work that we've pioneered.
But you do that, I've got to go now, but you do that with individual patients, so
Absolutely.
You only need the 3 to 4 days to do that. They're talking about, keeping these things alive so you can just continue to experiment on them.
To do interesting experiments.
Can't be a bad thing.
Oh, no, it's very good. It's a very interesting breakthrough, but I'm not sure that it's going to change individualization of patient treatments.
All right. So we're moving pretty quickly, but this is not maybe the breakthrough that I thought it was.
It's a breakthrough, and I think it's important, but we have work to do.
All right, all right. Dr. Nagourney, thank you for coming in. We appreciate it.